Good morning, people. Can you hear and see me? Anyone please respond? Huh? Raibag, Chikodi, connected? Please confirm me if you can hear me clearly. Ranabanur, can you confirm me if you can hear me? What is the distribution going on? What distribution is going on, Ranabanur class? No, but nobody is responding. Shivanand sir, can you hear me? Is class connected? Archana ma'am, Chikkodi and class 21 and class 22. I'm not able to hear you if anybody is speaking. Yes, Chikkodi class 21, I can see you, but no, I can't hear you. Check your mic. Can
Hello,
you? Yes.
21.
Got it. Class 22. Sudharani ma'am, class 22.
Hello. Yes, ma'am.
Please speak.
Hello.
Okay, class 22. Okay, fine. Raiba class 21, class 1. Kelly Raiba class 1. Shivanand sir, where is the mic? Hello, people. Okay, class 2. Raiba class 2. Raiba class 2, where is the mic? Ranabanur, how about you? Can you hear me?
Hello,
Raiba
madam.
class 2, Kelly Raiba class 2, please speak. Can you hear me? If you are speaking, I am not able to hear you.
Good morning, ma'am. Good morning, ma'am.
Good morning,
Which
ma'am.
class is this? Which class is this?
Chikodi 21.
Chikkodi 21, yes. Nim gotaito. Chikkodi, both the classes are joined. Wait, I am waiting for other batches. Hold on.
Good morning, ma'am.
Yes, who is this?
Rana Bennu.
Okay, Ranabanur class 2.
Only one class, ma'am.
Oh, one day class is there,
Yes,
Yes,
ma'am.
ma'am.
Okay. There should be 2, na? Okay, fine. Dharwad, you are also there?
Yes, ma'am.
Where is the
Yes,
mic
ma'am.
with you?
Yes, ma'am.
Dharwad, Divya ma'am?
Yes, ma'am. Good morning,
Okay,
ma'am.
connected, well connected. Okay, we have only 4 batches then. Okay, fine. Okay, how are you all people? Hello, how was your vacation? How was your exam?
We fine,
Fine,
ma'am.
ma'am.
Exam also
Fine.
fine, ma'am.
Ishtiyak
Good,
dull
ma'am.
idira, ishtella suti habba ella
Very
madidira
bad.
and still everybody's dull. Why?
Ma'am, are you fine, ma'am?
Yes?
Ma'am,
Say louder.
are you fine?
What? What is fine? Very small voice. ಇಷ್ಟು ಸಣ್ಣಗೆ ಮಾತಾಡೋದು ಕೇಳಿಸಲ್ಲ ನನಗೆ.
Ma'am, are you fine?
Yes, I'm also fine. Thank
Ma'am,
you.
ನಿಮ್ ಕೈಗೆ ಏನಾಗೈತ್ರಿ?
ಒಂದ್ ಸ್ವಲ್ಪ ಮೈಕ್ ಹಿಂಗ್ ಹಿಡ್ಕೊ.
Ma'am, ma'am.
ಟಾಕ್
ನಿಮ್
ಪ್ರಾಪರ್ಲಿ.
ಕೈಗೆ ಏನಾಗೈತ್ರಿ?
ಹೊಡೆದ್ ಬಂದೆ ಯಾರಿಗೋ.
Fracture.
I had a small accident. Okay, it's okay. Fine. So, okay, come back to the class. Dharwad people, Dharwad students, all your eyes on the board. Okay, ಏನ್ ಮಾಡಿದ್ವಿ? Do you remember what did we do? Ranabanur girls, all of you, good morning.
Good morning, ma'am.
That's like
Good
my
morning,
girls.
ma'am.
Good. What did we do before going to your vacations and exams? What did we do? Anyone? Which chapter are we doing, people? Chikkodi
Molecular
class
basis of
22.
inheritance.
Regular basis.
Chikkodi class 22. What's wrong with you all? What are you doing, class 22? Huh? First class boys ko bedri. Don't do that. ಹಾ ಏನು
Hi,
ಯಾರೋ
ma'am.
ಏನೋ ಹೇಳ್ತಾ ಇದ್ರು ಹೇಳಿ ಎಸ್ ವಾಟ್
Good
ಚಾಪ್ಟರ್
morning,
ಆರ್
ma'am.
ವಿ ಡೂಯಿಂಗ್ ಅಟ್ಲೀಸ್ಟ್ ಚಾಪ್ಟರ್ ಯಾವ್ದು ಮಾಡ್ಲಿಕ್ಕೆ ಅದನ್ನ ಅದನ್ನರೇ ಹೇಳ್ರು
Molecular basis of inheritance. Basis of
ಥ್ಯಾಂಕ್
inheritance.
ಯು ಥ್ಯಾಂಕ್ ಯು ಸೋ ಮಚ್ ಎಸ್ ವಿ ಆರ್ ಡೂಯಿಂಗ್ ಮಾಲಿಕ್ಯುಲರ್ ಬೇಸ್ ಆಫ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಆ್ಯಂಡ್ ವಾಟ್ ಇಸ್ ದಿಸ್ ಪೀಪಲ್ ಎನಿಬಡಿ ನೋಸ್ ದಟ್ಸ್ ಬೆಂಗಳೂರು ಪ್ಯಾಲೇಸ್ Okay, fine. So as far as I remember, we have to start with uh, search of genetic material, correct? Nan nenapiro prakara we have done nucleosome model, heterochromatin, chromatin, that nucleoid structure, basic first half first half alla first fourth 1/4 eno madidvi. Then we had to start from search of genetic material, right?
Yes, ma'am.
ರಿಮೆಂಬರ್ ನನಗೆ ನೆನಪಿದೆ ನಿಮ್ಗೆ ಯಾಕಿಲ್ಲ ಓ ಮೈ ಗಾಡ್ ಐ ಕಾಂಟ್ Okay, so to give you a quick revision, we have already completed about the portion which is consisting of chromosome structure or DNA length, then how a nucleosome model was there, how the solenoid model is there, heterochromatin, euchromatin, illivargu agittu, and we have solved questions also. The search of genetic material, that is the experiments. Which 3 experiments? Ranebandur class girls, all of you wait. I want only Ranebandur to answer, not focusing. Which 3 experiments? Search of genetic material. Books in ನೋಡ್ತಿರೋ midterm exam ಆಗಿದೆ ಅಲ್ವಾ? You had this chapter for your midterm exam, right?
Transforming principle.
Okay. First one is transforming principle. ಯಾರು ಕೊಟ್ಟಿದ್ದು? Come on, hurry up.
Griffith. Frederick Griffith.
Your voice is little bit shaking. Maybe check the connectivity or hold it properly. Okay, first Frederick Griffith gave the first experiment of transforming principle. Then
Hershey. Alfred Hershey. Hershey and Chase.
no, MacLeod McCarty.
Very.
This is the second experiment which gives the biochemical nature, or you
Characterization.
can say biochemical characteristics or biochemical analysis. Second experiment, correct? And then third one, Hershey and Chase.
And Chase.
Hershey and Chase gave a proof that
Genetic
what
material is
DNA
DNA.
is a genetic material.
DNA is a genetic material. Yes, then Hershey and Chase gave the experiment. So these 3 experiments gave you the, first of all, they are very important for your theory also, because theory alli baro chances ide, and you can expect from this chapter almost 11 to 14 questions. So be very very careful, okay? Don't miss this chapter. So let us see all these 3 experiments. Griffith gave the very first experiment somewhere around in 1928. Somewhere around in 1928. These people gave in 1933 to 1944. And the proof came around somewhere in 1953.
1952.
52, na?
Yes, ma'am.
Okay, 52. Okay, year, why am I mentioning year, you know? Because in NEET, CET ಅಲ್ಲಿ ಕೇಳೋ ಡೌಟು, ಬಟ್ NEET you may expect year also. ಸುಮ್ಮನೆ, because generally ಏನಾಗುತ್ತೆ, we do not pay attention to the year of the experiment. But NEET people are very wise. They can give you question year. So I just mentioned you. If I'm going wrong somewhere, please correct me. Okay, ನೋಡೋಣ experiments fast. Let us go through the experiment. Very first experiment. Oh. Yes. ಏನು ಹೇಳುತ್ತೆ ಈ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್? ವಾಟ್ ಡಿಡ್ ಹಿ ಡೂ ಗ್ರಿಫಿತ್? ಎನಿಬಡಿ ಕ್ಯಾನ್ ಎಕ್ಸ್ಪ್ಲೇನ್? ನಾನಂತೂ ಮಾಡ್ತೀನಿ. ಬಾಯ್ಸ್, ವೆರಿ ಲೆಸ್ ಸ್ಟ್ರೆಂತ್ ಟುಡೇ. ಧಾರವಾಡ ಬಾಯ್ಸ್, ಕ್ಯಾನ್ ಯು
Ma-
at
Ma'am,
least tell me what bacteria did Griffith
ma'am,
use?
boys are CS.
Streptococcus pneumoniae.
Streptococcus pneumoniae.
Correct. So he
Griffith,
used
Griffith,
Streptococcus
ma'am, Griffith
pneumoniae.
injected mice into the
Hmm?
Griffith injected mice into the R
Okay,
strain.
wait. Correct. Then there are two strains, which is rough strain
Hmm.
and
Smooth
Smooth
strain.
strain.
strain. Correct. Smooth strain is virulent. Virulent andre disease causing. Virulent, VIR I'm writing as. Virulent means the one which can cause the disease. Infection. ಯಾವ infection? Pneumonia will cause pneumonia infection, right? Streptococcus pneumoniae causes pneumonia simply. But our strain was not causing any infection. It was a healthy or a good strain, you can say. But what was there in a S strain that causes infection? S strain ಅಲ್ಲಿ ಏನಿದೆ that causes infection?
Fur coat.
Mucus coat.
The mucus coat,
Mucus.
the gelatin coat that is present over the bacteria, which
Polysaccharide
is
coat.
made up of polysaccharide. And because of that coat, the shiny layer or texture was seen, and ಅದಕ್ಕೆ ಆ ಸ್ಟ್ರೇನಿಗೆ ಸ್ಮೂತ್ ಅಂತ ಹೇಳ್ತಿದ್ವಿ. Whereas the lack of coat gave a rough appearance, so rough strain. Correct. So yes, first step was somebody explaining when you inject the rough strain,
When injected the rough strain,
the
mice
mouse
will
lives, which means
but the mice
rough
will
strain
lie.
is not causing any pneumonia or any disease. Simply, then smooth strain
Then
hakidre.
smooth strain will injected into the mice. Mice will die.
Who is speaking ಸ್ವಲ್ಪ loudly because voice is very small. ಸ್ವಲ್ಪ
Uh,
ಜೋರ್ ಮಾತಾಡು ಅಷ್ಟೇ.
yeah, strain injected into the mice, and mice will die.
Okay, so illi mouse dead because you are injecting the smooth strain,
Yes,
and
strain
this
or
is the
smooth
proof
strain.
that smooth strain are the virulent or disease causing. Then, what happened in
Means
the third step?
heat
What?
killed strain in heat killed strain is injected into the mice. Remain mice will alive.
But did anybody think why did he try to kill the heat skill? ಈ ಸ್ಟೆಪ್ ಯಾಕೆ ಮಾಡಿದ್ರು? ವೈ ಗ್ರಿಫಿತ್ ಥಾಟ್ ದಟ್ ಐ ಶುಡ್ ಗಿವ್ ದ ಹೀಟ್ ಕಿಲ್ಲಿಂಗ್ ಟ್ರೀಟ್ಮೆಂಟ್ and then put that bacteria into the mice. Any
Ma'am,
logic behind that? Think.
ಹೀಟ್ ಕಿಲ್ ಮಾಡಿದ್ರೆ ಅದ್ರೊಳಗಿಂದ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಕಿಲ್ ಆಗ್ತೈತ್ರಿ. ಸೊ ಅದ ಏನು ಎಫೆಕ್ಟ್ ಆಗಂಗಿಲ್ರಿ. ಅಂದ್ರ
Correct.
ಈಗ
So
ನಾವು
heat kill.
ನಾರ್ಮಲ್
Correct. Heat kill could weaken the bacteria. Sometimes not completely death of the bacteria. It can simply sometimes weaken the bacteria. So weak ಆಗ್ಬೋದು ಅಥವಾ completely kill ಆಗ್ಬೋದು, depends upon how long you are giving the treatment. Correct. So yes. So after heat killing, because there is no virulent smooth power is available to the S strain, therefore it was not able to cause any disease and mouse survived. So heat killing must have definitely done something with that S strain bacteria, due to which they are not able to do any disease to that particular mouse. Radamanur girls, ಇನ್ನೊಂದ್ಸತೆ ವಾರ್ನಿಂಗ್ ಕೊಡಲ್ಲ. I will ask the class to shut down. Then
and then
lastly,
rough strain and heat kill smooth strain injected into the mice. So mice will die.
correct. So what he did now, a fourth idea. ಈಗ ಸ್ಮೂತ್ ಸ್ಟ್ರೇನಿಗೆ ಹೀಟ್ ಕಿಲ್ ಮಾಡಿದ್ರೆ he found out smooth kill normal ಇದ್ದಾಗ smooth strain is dying the mouse, but heat kill smooth strain is keeping the mouse alive. So now what he wanted to check, to mix these two, mix the heat kill smooth strain and a rough strain, because what was his assumption, ಎರಡು ಕಡೆ ಮೌಸ್ ಲಿವ್ ಆಗ್ತಾ ಇದೆ. ರಫ್ ಸ್ಟ್ರೇನ್ ಹಾಕಿದ್ರು ಇಟ್ ಹ್ಯಾಸ್ ಟು ಲಿವ್, ಸ್ಮೂತ್ heat killed ಹಾಕಿದ್ರು it has to live, the mouse has to live. So then mixture of this should give you the mouse where it lives, but that was not the result. When he mixed the rough strain with the heat killed S strain, he could find that mouse is dying. Then what was his conclusion? Anyone from Dharwad, please? This chapter is done to you, Dharwad? I think this chapter is done to everyone, na? There is no batch ಅಲ್ಲಿ ಯಾರಿಗೂ ಮಾಲಿಕ್ಯುಲರ್ ಆಗಿಲ್ಲ ಅಂತ ಹಿಂಗೆ ಇರ್ಲಿಕ್ಕೆ ಸಾಧ್ಯ ಇಲ್ಲ ಇವಾಗ, ರೈಟ್? ಧಾರವಾಡ ಪೀಪಲ್ ಸ್ಪೀಕ್. What's wrong, Divya ma'am?
They are not saying answer.
I just
Hello?
asked whether this chapter is done or not. ಇದು
Yes,
ಮಾಡಬಾರ್ದ
ma'am.
ಆನ್ಸರ್? ಏನು ಅಂತದ ಇವರಿಗೆ? This chapter is done or not? My simple question.
Yes, ma'am. Yes, ma'am.
ಮತ್ತೆ still in a vacation mode? Huh? Come on, people. You are in PU2. We don't have much time. Buck up. Give strain to your brain now. Okay, no more time pass. We don't have time also to do a time pass now. Chikkodi class 21 boys. ಏನು
Ma'am.
ನಡೀತಾ ಇದೆ ಅಲ್ಲಿ? Class 21 girl, the one who is having mic. What is happening there? Class
Excuse me, ma'am. QR code distributed, ma'am.
ಏನು?
कि मैम QR code distribution ಮಾಡಾತಾರ ರೀ.
Okay. Okay, fine. Griffith experiment clear? Anyone, any doubt in Griffith experiment? Please ask now. ಆಮೇಲೆ ಮತ್ತೆ ಹೋಗಿ ಮುಂದೆ ಹೋದ್ಮೇಲೆ ಕ್ವಶನ್ ಮಾಡಬೇಕಾದ್ರೆ ಮ್ಯಾಮ್ ಅದು ಅರ್ಥ ಆಗಿಲ್ಲ ಅನ್ನೋಕ್ಕಿಂತ now you have a chance. Griffith experiment or transforming principle experiment, is it clear what conclusion he draws here? The conclusion in the last step is what? Something from the heat killed S strain was transforming the R strain into the virulence to make it virulent. Virulent means disease causing. Something was changing it. Transforming means something was changing it. But what? Still he doesn't know. Griffith said something is there which is changing the rough strain to a smooth appearance and making the disease. But what? Still Griffith was also not knowing. This gets then more clarity with second set of experiment, which is McLeod-McCarthy, where the biochemical nature was analyzed. Biochemical nature was analyzed andre ಏನು? Simply ಅದೇ Griffith experiment, the same Griffith experiment, they continued to check what actually the thing in cell is making it transformed. Protein, DNA, RNA, lipids, anything could be there which is making the conversion
chart is
from
not visible.
rough strain to smooth strain. So what they did, now it is visible. Is this little bit visible?
Yes, ma'am.
Okay, so what experiment McLeod-McCarthy or every McLeod-McCarthy anta ಹೇಳ್ತೀವಿ, these three people, they tried to treat the heat kill strain bacteria, S strain bacteria, with different enzymes. Lipids ಹಾಕಿ ನೋಡಿದ್ರು, lipids kill ಮಾಡಕ್ಕೆ lipase ಬೇಕು, protein kill ಮಾಡಕ್ಕೆ protease ಬೇಕು, then RNA kill ಮಾಡಬೇಕಂದ್ರೆ RNAs, to kill DNA. Kill means what? Simply to destroy or to remove. Let's say I have a test tube. In that test tube, I will just put protease enzyme. So, ಈ cell ಅಲ್ಲಿ ಇರೋ ಎಲ್ಲ proteins will get denatured. Proteins ಬಿಟ್ಟು ಮಿಕ್ಕಿದೆಲ್ಲ as it is, it will be there. Then in second set, if I have a test tube and I have added RNase enzyme, this will degrade only RNA. So RNA bit mattella constituents of cell will remain as it is. That is what, this is what the principle, or this is what the main basic fundamental thing was done in this experiment. What they did, they took different types of test tubes, added one type of enzyme in all the test tubes, and then infected again to the mice to check whether the mice is living or whether the mice is dying. So prati sate bere set of enzymes hakidaga, what happened when they introduced protease? The still mice was found dead, which means when the mouse is found dead, that means protein was actually nothing, which is not being the transferring factor. Similarly, do it with lipids, carbohydrates, RNA, every strain. But finally, when the one set of experiment where they used DNAs and this inactivated the DNA, then they found out that R strain were not getting transformed and the mice survived. Now, when the mice is surviving, which means R strain is not getting transformed to rough strain, and DNA is supposed to be the chemical nature which is transforming the heat kill S strain or the rough strain into the smooth strain. So, heat kill S strain tananta DNA property was giving it to the rough strain, so that now rough strain can produce the coat around it and cause the disease. ಇಲ್ಲಿ ಏನಾಯಿತು, ಇನ್ ದ ಫಸ್ಟ್ ಕೇಸ್ ಪ್ರೋಟೀನ್ ಕಿಲ್ ಮಾಡೋಕ್ಕೆ ಪ್ರೋಟಿಯೇಸ್ ಹಾಕ್ತೀರಾ, ಪ್ರೋಟೀನ್ ವಿಲ್ ಗೆಟ್ ಡಿಸ್ಟ್ರಾಯ್ಡ್, ಬಟ್ ಡಿ ಎನ್ ಎ ಹಾಗೆ ಇತ್ತು, ಹಾಗಾಗಿ ದ ಮೈಸ್ ವಾಸ್ ಸ್ಟಿಲ್ ಗೆಟಿಂಗ್ ಡೆಡ್. ಇನ್ ದ ಸೆಕೆಂಡ್ ಕೇಸ್ ಆರ್ ಎನ್ ಎಸ್ ಎನ್ಸೈಮ್ ಹಾಕಿದ ಮೇಲೆ all RNA from the cell will get destroyed, but other particles, other components are still like that only. So still it was found that when this mixture is injected to mice, the mice was dead. Means something is still there that is converting the rough strain to a smooth strain. Then when finally DNA's enzyme ಹಾಕಿದ್ಮೇಲೆ and then that mixture ಅಲ್ಲಿ ಏನಿದೆ, heat kill, smooth strain and rough strain. So when heat kill, smooth strain, rough strain and DNAs ಇವು ಮೂರು ಕಾಂಬಿನೇಷನ್ ಹಾಕಿದ್ಮೇಲೆ and injected to the mice, they found out that mice is now what, living or dead? Living, because
Right.
DNAs ಏನು ಮಾಡ್ತು, DNAs destroyed the DNA from the heat killed and R cells. So DNA destroyed ಆದಾಗ the mice was surviving, which means ಆ ಮಿಕ್ಸ್ಚರ್ ಅಲ್ಲಿ heat kill S bacteria DNA destroy ಆಯಿತು, that it could not transform the R strain to the S strain. And this was the first proof that protein and RNA is not a transforming principle, but DNA is a transforming principle ಅಂತ first evidence or first attempt ಇತ್ತು. Getting? Yes? Then the last proof that sure shot it is the DNA anta, lastly who gave this?
She and cheese.
Hershey and Chase experiment, right? What is the alternative name for this? One more name ide called as
Blender's experiment.
why blender, you know, literally blender was used, the mixture jar ಗೆ ಬ್ಲೆಂಡರ್ ಅಂತ ಹೇಳ್ತೀವಿ, ಅದನ್ನೇ ಲಿಟ್ರಲಿ ಯೂಸ್ ಮಾಡಿದ್ರು, ದಟ್ ಈಸ್ ವೈ ದಿಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಬ್ಲೆಂಡರ್ಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್. ಓಕೆ, ಎನಿಬಡಿ ಫ್ರಮ್ ಧಾರವಾಡ ಓನ್ಲಿ ಐ ವಾಂಟ್ ಟು ಎಕ್ಸ್ಪ್ಲೇನ್ ಮೀ ವಾಟ್ ಟೈಪ್ ಆಫ್ ಆರ್ಗ್ಯಾನಿಸಮ್ ವಾಸ್ ಯೂಸ್ಡ್ ಬೈ ಹರ್ಷೆ ಅಂಡ್ ಚೇಸ್? ಏನು ಯೂಸ್ ಮಾಡಿದ್ರು?
Bacteriophage and E. coli bacteria.
ಹಾ, ಏನು?
Bacteriophage and the E. coli bacteria.
ಕರೆಕ್ಟ್. ವಾಟ್ ಈಸ್ ದ ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್ ಅಂದರೆ ಅವ್ರದ್ದು ವಿಶೇಷತನ ಏನು? ವಾಟ್ ಅ ಬ್ಯಾಕ್ಟೀರಿಯೋಫಾಜ್ ಡಸ್?
Bacteriophage is the group of virus that kills the bacteria.
Correct. Bacteriophages are the group of viruses that infects the bacteria. Correct. T4 ಫಾಜ್ ತಗೊಂಡಿದ್ರು actually.
Ma'am, chat is not visible from Ranabinur. Screen is also blur, ma'am.
It's all blur.
Okay, fine, no problem. You have the chart in your book also. Fine, I will read it for you. ನಾನು ಓದ್ತೀನಿ ಬಿಕಾಸ್ ಇದಕ್ಕಿಂತ ಜಾಸ್ತಿ ದೊಡ್ಡದಾಗಲ್ಲ. ಐ ಕೆನಾಟ್. ನೋ, ಇದಕ್ಕಿಂತ ಜಾಸ್ತಿ ಆಗಲ್ಲ. ದಿಸ್ ವಿಲ್ ಬಿ ಬ್ರಲ್ ಅಲ್ಲ, ಇದೆಲ್ಲ ಕಾಣಲ್ಲ ನಿಮಗೆ ಅಲ್ವಾ? ಯು ಕೆನಾಟ್ ರೀಡ್ ದಿಸ್, ಐ ಗೆಸ್. I'll tell you but what it is. I'll tell you, no problem. Okay, fine. Initial of this experiment, Hershey and Chase, Martha Hershey and Chase, what they did, they have taken the bacteriophage in two sets. One set mele and one culture medium mele they have used P32 and ಇನ್ನೊಂದು ಮೇಲೆ what is this?
Radioactive isotope.
What is this P32 S35?
As per us, radioactive was as per us
Radioactive, correct.
32.
So one plate or one set of experiment where bacteriophage were allowed to grow in the P32 containing medium. ಈಗ bacteriophage structure ಹೇಗಿರುತ್ತೆ? The bacteriophages are made up like this, correct? This is called as protein coat. Remember, PU1 ಅಲ್ಲಿ ಆಗಿದೆ ಇದು biological classification. And inside the protein coat you have DNA or RNA, any genetic material, whatever the virus has, correct? So when viruses, these phage and viruses, if I say virus, please understand it is nothing but I'm talking about phage only. If this phage or if this phage is allowed to grow on P32, because phosphorus is a part of a DNA, all the phages here will get labeled with this P32. The DNA of this phages will get labeled with P32. Whereas phages which are allowed to grow on S35, this will get labeled with the protein coat, because protein has sulfur containing amino acid. Anyone can tell me example of a sulfur containing amino acid? You did not thought why protein is labeled with S35, why DNA is labeled with P32? Any logic? People, ನೀವು ಇಷ್ಟು ಶಾಂತ ಇಷ್ಟು ಡಲ್ ಆದರೆ ಇವನ್ ಐ ವಿಲ್ ಗೆಟ್ ಬೋರ್ಡ್ ಟು ಟೀಚ್. ಕಮ್ ಆನ್, ಬಿ ಇಂಟ್ರಾಕ್ಟಿವ್. ಎಷ್ಟು ಚೆನ್ನಾಗಿದ್ರು ಮೊದಲು, ಇವಾಗೇನಾಯಿತು? ಮಿಡ್ ಟರ್ಮ್ ಆಯಿತು ಆ್ಯಂಡ್ ಯು ಆಲ್ ಆರ್ ಡಲ್. ರಾಣಿಬೆನ್ನೂರು ಗರ್ಲ್ಸ್, ಚಿಯರ್ ಅಪ್. ಏನಾಗಿದೆ?
Ma'am,
ದೀಪಾವಳಿ ಫರ ಜಾಸ್ತಿ ಆಯ್ತಾ? ಹಾಂ, ಒಬ್ಬರಾದ್ರೂ ಕೇಳಿದ್ರ ನನಗೆ ಹೌ ವಾಸ್ ಯುವರ್ ದಿವಾಲಿ ಮ್ಯಾಮ್, ವಾಟ್ ಡಿಡ್ ಯು ಈಟ್ ಮ್ಯಾಮ್? ಇಲ್ಲ ಅಲ್ವಾ? ನಾನೇ
excuse me,
ಕೇಳಬೇಕು.
ma'am. Hello, ma'am.
Your voice is not clear if somebody is talking.
Ma'am, how was your Diwali?
ನೀನೇನು ಕೇಳ್ತಿ ಹೋಗೋ, ಬೇಜಾರಾಯ್ತು ನನಗೇನೋ.
Am I
ನಾನು ಹೇಳಿದ
audible?
ಮೇಲೆ ಕೇಳ್ತೀರಾ?
It's okay, ma'am. Hey, ma'am. Ma'am,
Ma'am,
please
am I
hear me.
audible?
Your voice is very low.
Am I audible now, ma'am?
Huh, who is this? Who asked me am I audible?
Sneha from Chikodi,
Class?
20.
Where is
Class
class 20?
20.
Cody.
There are only two classes, no? 21 and 22?
Ma'am, camera on
Ma'am,
actually.
Where is
20
the
number
third class?
camera on actually.
Okay, okay. I can't see third class. Okay, fine, fine, fine. Okay, Chikodi, I got your problem. Yes, tell me. You are audible, class 20. Chikodi, you are audible. You want to ask
Ma'am,
something? You want to say something?
I wanted to ask you what happened to your right.
Hmm?
What happened to your right hand?
My hand. ಹೇಳಿದ್ನಲ್ಲ ಯಾರಿಗೋ ಗುದ್ದು ಬಂದೆ, ಹೊಡೆದು ಬಂದೆ ಅಂತ.
ma'am, ಯಾರಿಗೆ ಹೊಡೆದಿರ್ರಿ?
ಆ?
ಯಾರಿಗೆ ಹೊಡೆದಿರ್ರಿ?
ಹಿಂಗೆ ನಿಮ್ಮಂಥವ್ರು ಹುಡುಗರು ಸಿಕ್ಕಿದ್ರು, ಕಾಡಿಸಿದ್ರು, ಎರಡು ಏಟ ಕೊಟ್ಟೆ. No, I had a small accident due to which I have injured myself. So it's paining. Please cooperate with me if I'm not able to write much.
Ma'am, you should have taken a leave.
ಎಷ್ಟು ಮೊದಲೇ
You
ಇದ್ದು
should have
ಹೋಗಿದ್ದೀರಾ
taken a,
ಅದ್ರಲ್ಲಿ?
you should have taken a leave.
And then who will complete your syllabus? ಬೇಡ, I'll try to do how much I can do it. I just want your cooperation. ಸಾಕು, ನೀವು ಅರ್ಥ ಮಾಡ್ಕೊಂಡ್ರೆ I'm fine. ಏನು ಕೇಳಿದೆ? What did I ask? ಹಾಂ, why DNA is labeled with P32 and why protein is labeled with S35? Anybody?
Ma'am, DNA is a polymer of nucleotide.
Hmm?
Hence,
So,
nucleotide contain phosphorus.
good job. Who is this?
Hence, from Dharwad.
Guys, I will tell you again, hold your mic properly, speak loud. ತುಂಬಾ
Ma'am, I am from
noise
Dharwad.
ಕಡಿಮೆ ಇದೆ.
I am from Dharwad.
Yes, Dharwad, I can hear you. Okay. So I got your answer of DNA. What is answer for sulfur?
I don't know.
Okay, fine. That is very honest and good. I appreciate. Anyone else knows ಈ ಕಥೆ ಯಾಕೆ ಮಾಡಿದ್ರು? Why phages were allowed to grow on sulfur containing 35 S35 radioactive? Boys, nobody is answering me. ಚಿಕ್ಕೋಡಿ boys, ಏನು ಮಾಡ್ತಾ ಇದ್ದೀರಾ? ಅರ್ಚನಾ ಮ್ಯಾಮ್, ಅಳಗ್ಯಾಡ್ಸ್ರಿ ಎಲ್ಲರಿಗೂ ಸ್ಲೀಪಿಂಗ್ ವಾಟ್ ಬಾಯ್ಸ್ ವೇರ್ ಇಸ್ ದ ಮೈಕ್ ಅವ್ರ ಹತ್ರ ಯಾರ ಹತ್ರನೂ ಮೈಕ್ ಇಲ್ಲ ಚಿಕ್ಕೋಡಿ ಪೀಪಲ್ ಬಾಯ್ಸ್
Girls.
ಓನ್ಲಿ ಒನ್ ಮೈಕ್
Two mics are there, ma'am. Ma'am, two mics are there.
ಓಕೆ ಓಕೆ ಒಂದು ಕೆಲಸ ಮಾಡು ಆ ಮೈಕನ್ನ ಆ ವಿಂಡೋ ಆಚೆ ಹೊರಗೆ ಬಿಡೋದು ಪಾಸಿಂದ ಪಾರ್ಸಲ್ ಮಾಡ್ಕೋತ ಥ್ರೋ ಇಟ್ ಔಟ್ ಓನ್ಲಿ ಬೆಸ್ಟ್ ವೈ ಯು ವಾಂಟ್ ಟು ಸ್ಕೋಲ್ಡ್ ಮೀ ಆರ್ ಫಸ್ಟ್ ಕ್ಲಾಸ್ ಇದೆ ಆಫ್ಟರ್ ವೆಕೇಶನ್ ಬಾಯ್ಸ್ ಪ್ಲೀಸ್ ಬಿಹೇವ್ ಡೋಂಟ್ ಗೆಟ್ ಸ್ಕೋಲ್ಡಿಂಗ್ಸ್ ಸಿಹಿ ತಿನ್ಕೊಂಡು ಬಂದರೆ ಸಿಹಿ ತಿಂದಂಗೆ ಇರ್ರಿ ಅದನ್ನ ಜೊತೆ ಸಿಂಪ್ಲಿ ಚಕ್ಲಿ ತಿಂದಂಗೆ ನಾನು ಬೈಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಮಾಡಿದ್ರೆ ಅದನ್ನು ಈ ಕೈನೂ ಉರ್ಕೋತೀನಿ ಅಷ್ಟೇ ಓಕೆ ಚಿಕ್ಕೋಡಿ ಕ್ಲಾಸ್ 22 ಬಾಯ್ಸ್
Yes, ma'am.
Dharwad boys, anyone can answer me this YS35. Adan answer Nanil kotte. I gave you the answer. Nobody paid attention. If you are speaking, I can't hear you. You know? No. Randamanur girls, can anyone try? I gave you the answer to this already. YS35, P32, okay, the student told me because DNA has a phosphorus in it, hangagi P32 radio label use madidru. Correct. S35 ಯಾಕೆ ಯೂಸ್ ಮಾಡಿದ್ರು? Boy, I'm not able to hear you. No, teacher, please, respective teacher, the boy who is speaking, he should hold the mic very properly and hing ಮಾಡ್ತಿರಲ್ಲ ಅದು ಕೇಳಿಸಲ್ಲ. Hold the mic, speak and then put down the mic. Fine, I don't have time now.
Protein present in disulfide.
Yes?
Protein present in disulfide.
Protein has which class is this?
Amino acids.
It has sulfur containing, correct. Protein has sulfur containing amino acids. Hangagi sulfur we can label it, which means ille here in this phage protein coat will be labeled in this set and protein and phosphorus DNA would be labeled in this set of experiment. Okay, illivargu I hope it is clear now. So now we have two phages which are differently labeled, allow it to infect to the host E. coli simply.
Hello, ma'am.
So yenagtittu, what is the nature of a phage? Suppose this is a E. coli.
Ma'am,
Phage or the infection agovaga, whenever the phage infects the host, phage will only give its genetic material inside. The protein coat always remains outside. Every virus, okay, prati virus ide thara behave ಮಾಡುತ್ತೆ whenever, whether it's a phage, whether it's an animal virus, plant virus, viruses tandu only DNA can send inside the cell. Protein coat ಅಥವಾ capsomer ಅದೆಲ್ಲ ಒಳಗೆ ಹಾಕಕ್ಕೆ ಆಗಲ್ಲ. That is the normal nature of a virus. And what it does, DNA ಒಳಗೆ ಕೊಟ್ಟ ಮೇಲೆ, suppose this is the main DNA of the E. coli, the DNA of virus will go and insert itself into the bacteria's DNA. So ಏನಾಗುತ್ತೆ, after some time you can see this DNA will have a small particle of virus DNA also. This is called integration of the viral DNA into the bacterial genome itself. This is the nature how virus infects. Even if you talk about HIV virus in humans or retroviruses, any virus, ಯಾವ್ದು ಬೇಕಾದ virus ತಗೊಂಡ್ರೂ virus will give only genetic material inside, ಹೊರಗಿನ ಕೋಟ್ ಎಲ್ಲ ಆಚೆ ಹೊರಗಡೆ ಇರುತ್ತೆ. That is the thing. So ಈ ಥರ nature ಇರೋದ್ರಿಂದ the blenders experiment was very easy. What they did, same thing, here they have used the phosphorus containing phages and allowed the bacteria to infect. Here they have taken S35 containing phages and allowed the bacteria to infect. On the ಎರಡು ದಿನ ಆದಮೇಲೆ after infection in the blender they have separated the phage and E. coli cells. ಸೇಮ್ ಇದರಿಂದ ಫಾಸ್ಟಾಗಿ ತಿರುಗಿಸಿದ ಮೇಲೆ ಏನಾಗುತ್ತೆ, ಈ ಫಾಜ್ ಏನು ಮೇಲ್ ಕೂತಿರತ್ತಲ್ಲ, ದಿಸ್ ವಿಲ್ ಗೆಟ್ ಸೆಪರೇಟೆಡ್ ಆ್ಯಂಡ್ ಈಕೋಲೈ ವಿಲ್ ಗೆಟ್ ಸೆಪರೇಟೆಡ್. ನಾವು ಇಮ್ಯಾಜಿನ್ ಫಾಜ್ ಒಳಗಡೆ ಡಿ ಎನ್ ಎ ಇಲ್ಲ, ಡಿ ಎನ್ ಎ ಎಲ್ಲ ಒಳಗೆ ಹೋಗ್ಬಿಟ್ಟಿದೆ. ಓಕೆ, ಸೊ ಲಾಸ್ಟ್ಲಿ ಆಫ್ಟರ್ ಬ್ಲೆಂಡರ್ಸ್ ಸ್ಟೆಪ್ ಸೆಂಟ್ರಿಫಿಗೇಷನ್ ಮಾಡ್ತೀವಿ. ಸೆಂಟ್ರಿಫಿಗೇಷನ್ ಮೀನ್ಸ್ ಆ ಟ್ಯೂಬನ್ನು ವಿ ವಿಲ್ rotate it at a very high speed, RPM, if you know what is RPM. So when you rotate it at the RPM, that time what happens, the upper layer which is called as supernatant and the lower thing which is deposit is called as pellet. So P32 ಅಂದರೆ ಪ್ರೋಟೀನ್ ಹಾಕೋ ಎಕ್ಸ್ಪೆರಿಮೆಂಟಲ್ಲಿ ಪೆಲೆಟಲ್ಲಿ ಏನು ಕಾಣಿಸ್ತು, ದೇ ಕುಡ್ ಸಿ ದ P32. ಇದ್ರ ಮೇಲೆ ಎಕ್ಸ್ರೇ ಬಿಟ್ಟರೆ ದಿಸ್ ವಾಸ್ ರೇಡಿಯೋ ಲೇಬಲ್ಡ್, ದಿಸ್ ವಾಸ್ ಶೈನಿಂಗ್. ಲೇಬಲ್ಡ್ ಏನಿತ್ತಲ್ಲ ಇದೆಲ್ಲ ಗ್ಲೋ ಆಗ್ಲಿಕ್ಕತ್ತಿತ್ತು. ದಿಸ್ ವಾಸ್ ಗ್ಲೋಯಿಂಗ್. ಪೆಲೆಟ್ನಿಂದ ಒಂದು ಗ್ಲೋ ಬರ್ತಿತ್ತು ಬಿಕಾಸ್ ಆಫ್ ದ ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ P32. ವೆರಾಸ್ ಇನ್ ಸೆಕೆಂಡ್ ಸೆಟ್ ಆಫ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಏನಾಯಿತು, ಪೆಲೆಟಿಂದ ಏನೂ ಗ್ಲೋ ಇಲ್ಲ. ಯಾಕೆ? Because all the viruses were in this supernatant, and this suggests that the protein coat remained outside and the DNA was the only one that comes inside the cell of a bacteria, and which was the actual transforming property or transforming DNA. This is how they proved. ಇದು ಯಾಕೆ ಪಾಸಿಬಲ್ ಇತ್ತು ಗೊತ್ತಾ? ಬಿಕಾಸ್ ದ ವೈರಸಸ್ ಹ್ಯಾಸ್ ದ ಕೆಪಾಬಿಲಿಟಿ ಟು ಓನ್ಲಿ ಸೆಂಡ್ ದ DNA inside. ಪ್ರೋಟೀನ್ ಕೋಟ್ ಹೊರಗಡೆ ಉಳಿಯುತ್ತೆ. ಬಿಕಾಸ್ ಆಫ್ ದಿಸ್ ಪ್ರಾಪರ್ಟಿ ದ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ವಾಸ್ ಸಕ್ಸಸ್ಫುಲ್ ಆ್ಯಂಡ್ ದಿಸ್ ಗೇವ್ ಅ 100% ಪ್ರೂಫ್ ದಟ್ DNA is the only transforming principle. Yes? Question solve ಮಾಡೋಣ if you all thorough with this. Yes or no? You want
Yes,
me to repeat
ma'am.
anywhere?
Yes, ma'am. Yes, ma'am.
Ma'am.
Huh. Try doing this question. Raise the cards. Raise the cards. Don't answer me. Raise the cards.
Option C.
Hey, nanin ಹೇಳಿದೆ?
C, ma'am.
This bacteriophage. Don't answer me. Raise the cards. It's easy question. Okay, I have answer as from Chikkodi. Okay. Okay, most of you are answering 3, is it? When a bacteriophage
Yes, ma'am.
bacteria, only genetic material was first proved by Hershey and Chase. Okay. Everybody goes with this? Any doubt here?
No, ma'am.
Ma'am.
Again, raise the cards. Please do not disclose the answer. ನಾನು ಹೇಳಂದಾಗ ಮಾತ್ರ ಹೇಳಿ. For this, again, raise the cards.
Hey.
Eh? You don't get my instructions or what? Okay. Harvard has the cue card?
C, ma'am.
Okay.
C, ma'am. E, ma'am.
Again C, but there is confusion in A and C both. I am getting answers A and C both. Few of them are answering A also. Heat killed and life type pneumonia bacteria was injected into the mice. The mice will very direct question. You are taking C. Di and S type bacteria will be recovered from mice. Okay. And here di and R type Allah. A is wrong. Na R type could not be recovered. R has been transformed or changed itself into the S type. So this is more appropriate. Yes? Or do you think the fourth option? Read this. Fourth option, do you think so? Okay. Anybody has any doubt here? Shall I go ahead?
Yes, ma'am.
Okay, someone tell me this question directly now. No cards, tell me directly.
1928. C, ma'am.
Option C.
Okay. Try this. Raise the cards. Try this.
First,
Bit
ma'am.
confusing. Bit
Option
confusing.
A.
Option A. Okay, which
Option
class
D.
is option A?
A, ma'am. Ranebennur
Option
from.
A from Dharwad.
Ranebennur, the all of the girls of Ranebennur do not participate. I can see the one who are sitting towards the last window. That rows much are not participating. Where is the mic in Ranebennur class? Mic ಯಾರ ಹತ್ರ ಇದೆ?
Yes,
Parcel.
ma'am.
Pass the mic towards your right. Pass the mic to your right. Last row. Ali last row ಹೋಗ್ಲಿ. Last bench alla, last row. Anyone from that last row talk to me. Yaradru? 1, 2, 3, 4, 5, 6 benches are there. Answer this. Yake mic tagondro? Give the mic. Girl, Ranebennur, give the mic to them. Last bench al row, last row. Nimage artha
Option
agata?
A,
This
ma'am.
is row. Okay. Who is
Option
there?
A, ma'am.
Where is the mic? Pass. Pass to your right. From that whole column ಅಂತ ಹೇಳ್ತಾರೆ ಈಗ ಉದ್ದುದ್ದ ಇಟ್ಕೊಳ್ಳಿ ಕಾಲಮ್ ಓಕೆ ಯು ಡೋಂಟ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ರೋಸ್ ಆ್ಯಂಡ್ ಕಾಲಮ್ ಅಲ್ಲಿಂದ ಫ್ರಮ್ ದ್ಯಾಟ್ ಕಾಲಮ್ ಎನಿಬಡಿ ಆನ್ಸರ್ ಮೀ ಗರ್ಲ್ಸ್
Option A, ma'am.
ವೇರ್ ಇಸ್ ದ ಮೈಕ್ ಆ್ಯಂಡ್ ಹೂ ಇಸ್ ಆನ್ಸರಿಂಗ್
Option A, ma'am.
ಹೂ ಆನ್ಸರ್ಡ್ ಯು ಸುಮ್ಮನೆ ಅವ್ರು ಹೇಳ್ತಾರಂತ ನೀನು ಹೇಳ್ತೀಯಾ ನೋ ಓಕೆ ಅದರ್ಸ್ why option A? Why you think idalla? Or 2 and 4 also, why not? Hurry up people, hurry up.
DNAs affect the transformation.
Correct. So DNAs affect the transformation hangagi D and B tagad bidana. Idardi ಏನು ಕಥೆ? What about this? NCERT ಅಲ್ಲಿ ಕೊಟ್ಟಿಲ್ಲ ಮ್ಯಾಮ್. NCERT ಅಲ್ಲಿ ಪ್ರೋಟೀಸ್ and RNS ಕೊಟ್ಟಿದ್ದಾರೆ. So we go ahead with it. Well, that's correct also. Actually lipids ಬರಬೇಕು. Lipase also does not affect, but there is no such statement in your NCERT. Technically lipase will also not affect the transformation. Practically ಹೋದ್ರೆ, but ಈ ಥರ NCERT ಅಲ್ಲಿ ಮೆನ್ಷನ್ ಮಾಡಿಲ್ಲ. That's why go ahead with this clearly. ಇದು
Option A. Option A.
okay.
Option A, ma'am.
Okay.
Option A. Option A, ma'am.
S35 was allowed and then agitating in the blender ಅಲ್ಲಿ ಹಾಕಿದ್ಮೇಲೆ virus and E. coli got separated after centrifugation, then radioactivity ನೋಡಿದ್ಮೇಲೆ radioactivity means the glowing activity was found in
Option A, ma'am. From Ranebennur.
why not B? ರೇಡಿಯೋ ಪೆಲೆಟ್ ಅಲ್ಲಿ ಇತ್ತಲ್ರೋ ಅವ್ರ ಸೂಪರ್ನೆಟೆಂಟ್.
The protein codes are
ರೇಡಿಯೋ
not
ಆಕ್ಟಿವಿಟಿ ವಾಸ್ ಸೀನ್ ಇನ್ ಪೆಲೆಟ್ ಆರ್ ಸೂಪರ್ನೆಟೆಂಟ್?
Supernatant. Option A.
Why not pellet then?
Hello? Hello, ma'am.
Yeah, I can hear you. Option A, I understood. Why not pellet ಅಂತ ಕೇಳ್ತಾ ಇದ್ದೀನಿ.
Because the protein codes are lighter and
Correct, because they are using the S35, simple logic. This was the second set of experiment. First set of experiment where P32 is used, avaga radioactive would be in pellet. PP, SS, simple logic. S35 iddaga you will see it in supernatant, pellet iddaga phosphorus 32. Simple logic. It is a very direct answer. Please use your brains. Okay. Then do this. Easy again. Anyone?
Option D, ma'am.
Option D, ma'am.
Confusing between A, C and D. Yake the process of transformation is not affected by which of the following?
Option A.
All three barbecala RNase, lipase and protease. Huh? Which is not affected anta kehta idaro.
Option A, ma'am.
DNA is not affecting.
No, ma'am.
DNA is hakodrinda yenagate, DNA will get dyed and then there is no transformation. So which is the one which is not affected by transformation, which is not affected by any of the following? Which of the following?
Option A, ma'am.
Sure?
Yes, ma'am.
Okay. Fine. So what is next concept after this experiment? Search of genetic material, then RNA world, right? What are the properties, criteria to be a genetic material? What are the criteria or the properties? Genetic material.
It should be stable.
Okay, it should be
It should
stable,
be
structurally and chemically both.
chemically.
Should produce its replica.
Okay, then
Application and application.
it should be able to have or it should be able to do the mutation, show that there is a scope of evolution. Mutation adre tane variations or changes agatte, and when there are changes happening the organisms can evolve, it can grow. True. Then
Generate its own
It's
replica.
you know
Mendelian's
It should generate
characters.
its own replica, ma'am.
that should be actually first point. It should be able to replicate. And Mendel's point, they should follow the
Spread
Mendel
in the form of Mendelian's characters.
laws. Mendel laws follow ಮಾಡಬೇಕು. Correct? These are the four. Actually replicate should come first, then 1, 2, 3 and 4. I think this is the sequence. Okay, anyways, so do you think that DNA satisfies all these four properties? Yes, DNA is more structurally stable than compared to RNA. Why? What does the DNA has and what does the RNA has?
DNA has
The difference between the purine and pyrimidine. RNA has a uracil whereas DNA has
Thymine.
thymine. Then the 2-OH group of RNA makes it more unstable, or you can say makes it more reactive. Also, RNA cannot make its own replica and therefore cannot undergo any kind of mutation also. Right? So this was the reason due to which You cannot keep RNA as a genetic material. It is not following all the criteria. Mendel's laws ಅಂತೂ ಫಾಲೋ ಆಗೋದೇ ಇಲ್ಲ, ಕರೆಕ್ಟ್? ವೆರಾಸ್ DNA, where uracil was replaced by thymine, due to which the DNA became more structurally stable. Second, DNA has two complementary strands, right? This complementary strands gives the more stability, whereas RNA is single stranded. A single strand irodrindano it becomes more reactive, and this becomes less reactive, and less reactive, which means more stable. So when it is more stable, it can pass on from one cell to another cell, another cell to another cell. So pass on from the generation becomes easy, and the structure of chromosomes or the factors or the genes was actually found on DNA, which means it was following the Mendel's laws also. That's why we say the DNA could follow all these four properties, and DNA was acting as a genetic material. But then there was also a concept that DNA ಬರೋಕ್ಕಿಂತ ಮುಂಚೆ RNA ಬಂದಿದೆ ಅಲ್ವಾ? That's why we say it is a RNA world. Why you think in the process of evolution RNA is the first came and then the DNA? Again, answer lies in the backslide.
Which one mutates faster, RNA or DNA?
Which one? What faster?
Mutates faster.
RNA.
Mutation happens.
RNA. Are there reasons illi? Why you think RNA? RNA world, what do you study here? Under the RNA world we study RNA came first, and RNA indane DNA bandide anta we proof. There is a evidence RNA indane DNA bandide. So ಮೊದಲು RNA ಯಾಕೆ ಜಾಸ್ತಿ ಇತ್ತು? ಬಿಕಾಸ್ RNA ಹ್ಯಾಸ್ ಅ ಎಕ್ಸ್ಟ್ರಾ OH group, 2' OH group at the second carbon, and it is catalyst. RNA was acting as a catalyst. Andre, what is catalyst? The one which enhances the rate of reaction is catalyst. So RNAs were sometimes acting as a catalyst also. Main function of RNA is what? To prepare protein. mRNA indana protein ಮಾಡಬೇಕು. That is the main function. Apart from that, it is also acting as a catalyst. So RNA world ಹೇಳೋ ಉದ್ದೇಶ ಏನಂದ್ರೆ, lastly the ultimate goal of a cell or of a DNA is to produce protein, and this production of protein is possible only when RNA comes into picture. And then it was found out that evolution, if you have studied evolution, that very first molecule that came into the water because of the reactions happening was RNA. RNA unstable irodrinda and highly reactive irodrinda, it was not able to give from one cell to another cell stably. So when it is not able to pass itself from one cell to another cell, inheritance, that is passing on or genetic material property, is not been followed. That's why RNA, due to again in nature's law, lost its oxygen group. So one oxygen kadme agodrinda DNA ge ನಾವು deoxy ಅಂತ ಹೇಳ್ತೀವಿ. We say it is deoxy, removal of one oxygen from RNA. RNA is what? Ribonucleic acid. DNA is what? Deoxyribonucleic acid. RNA has actually two oxygen at 5th and 2nd position. This is the ribose sugar. Illi ಒಂದು oxygen ಇರುತ್ತೆ for RNA. But when this was removed, then we got DNA. ಇಲ್ಲಿ ribose sugar, you have phosphate here, and you have nitrogen base here. ಈ ಥರ. Then uracil, conversion of uracil to thymine was also one more addition to give the stability, and the complementary strand, single stranded, was now converted to double stranded, that is by reverse transcription, and this three, what you say, criteria gave the birth or gave the origin of DNA. But RNA world suggests what? DNA ಕಿಂತ ಮುಂಚೆ RNA ಬಂದಿದ್ದು, but RNA, because of its lack of ability to being stable, to have mutation and all, it had to convert to DNA, and DNA was now able to give it to different cells stably. So we said DNA is a genetic material. That's it. Yes?
Yes, ma'am.
Okay, few quick questions on this because this is a very, very easy concept. Try. Try. You can directly tell me. RNA can directly code for the synthesis of?
Proteins.
Option B.
Obviously, proteins, na? You have to have a proteins.
Option B. Proteins.
DNA, gene and codon agalla. They are going to give you proteins, and that's why they are able to express the characters. Correct. This is Griffith experiment. You can do it on your own. You do Griffith experiment ಇದೆ. I mean experiments ಇದೆ. Try this. People ಕೆಲವೊಂದು ಕ್ವಶನ್ ನಾನು ಸ್ಕಿಪ್ ಮಾಡಿದ್ದೀನಿ because they are easy. They are experiment based question. You have to do it on your own. I'm giving it as your homework. Few easy questions. When you get this PDF, try to do it. If you don't understand, you can come and ask me. Do this. Nucleoside is A plus B. Nucleotide is this plus C. DNA is dash dash plus phosphate. What could be the answer? It's easy actually.
Option C.
C. Okay.
Yes.
Base. A is base. B is sugar. Correct. Base plus sugar gives you nucleoside. Then phosphate is C. Nitrogen is base plus pentose sugar plus phosphate gives you nucleoside. Correct. Then D plus E plus phosphate. Base plus sugar plus. Correct. Yes. Okay. Then this one. Quickly.
Option
D.
Option D.
Option
ma'am.
D, yes, we just saw because of all these three properties, DNA will resist the change to happen. Okay, fine. There are few more questions, people. There questions illi, which is again same thing kind of question. Because the concept is very tiny, I am leaving these questions to you. You only solve this when you get the PDF, and if you find any doubt, you can come back to me. I'm going to start with replication tomorrow. Do not miss the class. I'll meet you tomorrow, and please read and come. This chapter is very big and important. Replication and transcription eradu. Read and come. Okay?
Bye,
I'll stop here.
ma'am.
Do the question, okay? Left out questions ಮಾಡಬೇಕು ನೀವು. Chalo. Have a nice day, people. See you.
See you,
Bye.
ma'am.
Bye.
Thank you, ma'am.
Thank you. Give the feedback quickly.
Hello.